Okay, should be recording. So again, just like normal, I think y'all probably gotten the hang of it now. Again, on Blackboard, when these are done and recorded, after they download, I'll get them posted on Blackboard. So again, for those that need to, you can go back and review. If you haven't watched them, you can watch them. You can pause them, do all that kind of stuff. So everything you need, I guess, should be available. I asked you while we were gone to work on visible body and wander through the cell chapter and the practice quizzes. All right, so there's a couple of updates in that regard. What I'm going to do for a lecture today is I'm going to wrap up cells, basically try to hit some highlights that visible body talked about, maybe add a little bit of additional information that I think is relevant, pertinent as we move forward towards your first exam. I'll discuss that in a second. And then couple of other things. I have posted a Blackboard quiz. So, sorry, hold on, let me admit someone. I have posted a Blackboard quiz. So when you log into your Blackboard class shell, when you go to the content area and scroll down, you should see a folder that says exams and quizzes. If I remember correctly, I set it to open at 8 o'clock. I think it's set to open at 8, so it should be available now. It is that Blackboard quiz. Again, a couple things different from the visible body. So this is a Blackboard quiz. It is loaded in Blackboard. There are 10 questions. It's worth 10 points, and you have a 10-minute timer, and you can only access it one time. So once you open the quiz, you have to complete it in that sitting. So you don't have other access to it. It is just due by the end of the day, Sunday. So get that quiz completed before midnight. Don't start it at midnight. Again, if you started at 11:59 on Sunday, it's going to kick you out. It'll close. So again, take that quiz, get it out of the way. That's a Blackboard quiz. It's loaded under exams and quizzes. Again, you only get one opportunity for that quiz. So make sure you understand the information. It's in regards to terminology. Make sure you've practiced the terminology. The quiz is multiple choice and true false questions. So I think it's pretty straightforward in this first one. So just make sure you get that quiz and get that out of the way. One important thing about the quiz, you will be able to see your grade. You should be able to see your grade right after you complete the quiz, but you cannot view and check right and wrong answers right after the quiz. All right. The quiz is not made available until everyone has completed the quiz. That's just so that someone can't take it, get all the answers, and give them to everybody else. Not that you would. But the point here is you are not allowed to review your quiz until after everybody's completed it. So with that in mind, it basically means, assuming everybody completes it by the end of the day Sunday, you should then on Monday be able to review your quiz. If you click on it in the grade book, after everybody's completed it, then it'll let you see your questions. It lets you see the question, it lets you see your answers, and it lets you see any wrong answers or corrected answers. So if you have questions about it once you take it, if you're something's wrong and you think it was right, let me know. Again, I have made mistakes in uploading and writing questions and all that kind of stuff. So again, you can see your grade right after you take it, but you can't view it till everyone's completed it. That's Blackboard things. Okay, so that's the first thing. Make sure before Sunday you've completed your Blackboard quiz. All right, what I'm also going to do is post a visible body graded quiz. Right now, all that's available is practice quizzes, and the practice quizzes on visible body are for you to essentially do as many times as you want, as many times as you can, to essentially learn all the information and get a perfect score. What I'm going to do is after lecture today, I'm going to post a graded quiz. All right, it'll be some of the same questions, maybe even all the same questions. The point here is your graded quiz is for a grade, just like Blackboard. It is not loaded on Blackboard though. You access it via visible body. All right, so in the visible body module, after today, there will be a graded quiz. I will leave that graded quiz open until Sunday as well. All right, unlike the practice quizzes, you only have one opportunity. So you get to take that graded quiz one time. So make sure you're prepared. It again will be a 10-point quiz. So after this week, of all those 10-point assignments, you'll already have 2. Okay, so again, working towards getting the number And if I give you extra, if I can get more than the 10 that's required or whatever, we'll use those as bonus. All right. So there is a Blackboard quiz loaded on Blackboard. And after today, there'll be a visible body quiz. It's graded as well. Here's one of the differences. Your Blackboard quiz, the grade automatically shows up in your grade book. When you take it on visible body, it turns out that it shows up in your visible body grade book. I have to manually then go and upload those to Blackboard. I will get around to that probably next week after everybody's taken that graded quiz on visible body. So give me some time there. All right, you should though, after you take it on visible body, be able to view your quiz in that regard. So that's a little bit different than Blackboard. So again, 2 assignments, 2 things that are graded this week, a Blackboard quiz in Blackboard, and you have a visible body one I'll post after lecture today. Those are your first 2 10-point assignments. All right, they should be pretty straightforward. You should be able to get hopefully most of your points if you've prepped that way. Okay, so that's what you got. Now, my other plan is very quickly here in a second to wrap up the information on cells. I wanted you to work through visible body. I'm going to hit some highlights. So anything additional I give you today is also fair game. All right, everything in visible body is fair game. That's again why I've given you the practice quizzes over there and the graded quizzes to assess your knowledge base on that information. Because on Tuesday, next week, I don't know the date off the top of my head, but Tuesday, I think that is going to be June 23rd. Tuesday, June 23rd is your first exam. It is worth 50 points. It will be basically a bigger version of the Blackboard quiz. It will be 25 questions, 2 points each, a mix of multiple choice and true false. And it will cover everything up through the cell stuff that we've done in lecture. So everything will be fair game up through cells. All right. With that in mind, make sure you've asked questions if you have any before Tuesday. Make sure you've reviewed. What I'm going to do is post that exam for Tuesday. All right. That would be my goal. And I will give you till the end of the day, Wednesday. If people need additional time, you need to send me an email. All right. Exams, unlike quizzes, aren't basically you get all week long because I need to start new information then on the 25th, which would be Thursday. All right. So trying to keep us up to on pace on that. My goal for Tuesday would be to post an exam, and I'll let it close on Wednesday, and hopefully everybody can take it. If you have or need some exception, let me know. But 2 days, 2 complete days should be plenty, I hope. Again, the exam should not take you more than, there'll be a timer on it. And like I said, 30-minute timer. It shouldn't take you very long to complete it once you get the hang of that. So again, that'll make more sense too after taking a Blackboard quiz and seeing sort of the format of this. So we're kind of easing in slowly. Again, this may be different than some of your other classes, but you'll get an idea of the type of questions we're asking, what you're responsible for, and create a good foundation, not a ton of points before we get into some of the bigger exams. That would be my goal. All right. So what I'm going to do, and this is another important thing, so hopefully you're watching the videos. Tuesday, because we have an exam, I will not lecture. So if you look at that syllabus, you'll see that all that's posted on Tuesday is an exam. Again, the exam will leave open till the end of the day Wednesday. But again, we're not lecturing on Tuesday. So again, if you have questions about anything between now and Tuesday, just reach out. It would be great if you're prepared, and then Tuesday during the normal lecture time, take your exam, get it out of the way. So that would be sort of my goal. All right. So if y'all can do that, that would be fantastic. And again, if there's questions, concerns, just reach out, shoot me an email or anything like that, or I'll stay online here at the end of this video to see if people have questions. All right. So that gets all the nitty-gritty stuff out of the way, the assignments, what's coming up, and all that kind of blah, blah, blah. If there's not any questions, I forgot. Hopefully nobody's chatting. Sorry, I forgot to open my chat window. Will the visible body graded quiz be based on the multiple choice part or the dissection part like the practice quiz? Your visible body graded quiz is mostly on that multiple choice portion of it. All right. But technically speaking, if I go in there, the one I've created so far for visible body is mainly on that multiple choice part. All right. I think you should work on both. All right. And I actually may go in, and let me make this clear as well. So that's a good question. Right now, I want you to work on both parts, and you should. The graded quiz, mostly for the most part, is that multiple choice portion of it. But I want you to work on both parts because the exam that I write for Tuesday may pull information from both sections. So make sure you've at least gone through it. All right. Whether I ask questions about it or not, it's fair game. And so what I can promise you I will do, not the entire test, but I will pull some of the visible body questions for use on the exam. And that includes if I can get the pictures to download the dissection part, or at least a written question that refers to the dissection part, if that makes sense. My point here is I want to use visible body to supplement what we do in lecture. And so I think it should be fair that you should see some of those questions on your bigger exams. Otherwise, I think it's just wasted time. So I will pull a few of the questions on all of the exams you have, the 50-point or 100-point exams. Some of that will come from visible body. So it is in your best interest to do that visible body stuff, because again, these are basically saying if you've done that information, if you've done those assignments, those are basically like free points. All right. So you're supplementing your test that way. So please do both parts, but the visible body quiz for the most part focuses on that multiple choice portion. Okay. All right. Now that I got all my ducks in a row, I think. Okay, so go to share the screen here. Okay, what I asked you to do on your own, essentially use visible body and work through the cell and the cell membrane. And it highlights a bunch of things in that regard. It introduces cells, and much of that introduction to cells is probably things you've had back to high school biology, if you will. If not, and visible body does a nice job of wandering through, again, both the dissection of cells, these organelles and their functions, as well as the plasma membrane. It shows some good visualizations to go hand in hand with that. Again, just making sure we understand, I am planning on, after we finish up this section on cells, I am going to start on the tissues. It is not on exam 1 material, which is the 50 points. Ends here. All right, I am going to hit some highlights right quick on what I want to make sure we understand and recognize in regards to the plasma membrane, organelles, and the resting membrane potential. But as soon as I get through that, that is where your first exam starts. I am going to start new information on tissues today, but I want to get some of this out of the way. So, all right, we've got cells. Again, when I talked about cells and the study of cells, cytology. Ology means the study of, cyte means cell. So anytime you see that word, it's referring to essentially cells. Cells, the smallest unit of life. All right, it means it can perform all of the functions necessary for living things. All right, you are a multi-celled organism, and you have collections of cells, and those cells then are organized into tissues. Those tissues then into organs, into organ systems, and then into you, the organism, the human. All right, but you are a collection of cells. What's neat about this to me is your cells have all of some of the same basic parts. All right. So your cells have all of the same basic parts. The one you're probably most familiar with, your cells have a nucleus. Your cells have a cell membrane. Your cells have organelles. All right. Your cells have a cytoplasm, a fluid within them. There's an internal environment. There's an external environment, but all cells, no matter what kind it is, has all of these same basic parts. And one of those things that it revolves around is having a plasma membrane. All cells have a plasma membrane. Sometimes you see this called a cell membrane. All right. Now, what I think is unique, and the reason I bring this up is I think you can recognize that your cells that occur in your body are very different as well. And even though they have some of the same basic parts, they also have unique characteristics that differentiate them. In other words, you have muscle cells. And what does a muscle cell do? It simply contracts. A muscle cell is built so that it contracts. Why does it contract? It contracts for movement. I hope you recognize that a muscle cell is not the same thing as a bone cell. Muscle looks very different than bone. All right, or a nerve cell, if you will, or a blood cell. And what I'm trying to get you to recognize is all of those things are cells, and all of them have some of the same basic parts. But as we go through this class, we'll also highlight what makes them unique. How do you differentiate them? And a lot of that is based off then unique structures that they have. All right. All of your cells have a plasma membrane. Visible body wandered through that, and hopefully you were able to see that for simplistic purposes, your plasma membrane, your cell membrane, is composed of parts. All right? Your cell membrane as a whole is generally described as something called a fluid mosaic model. It is the best description we have of a cell membrane. Now, what that essentially means is, and I introduced this the other day, you have a cell. My black line here represents the cell membrane. Now we have stuff outside the cell, and I talked about that the other day when we talked about the water environment, your extracellular fluid. We also have that fluid inside the cell, the intracellular fluid. Back in your basic biology class, you may have called that your cytoplasm, the fluid inside your cell. All right? So we have a cell membrane. The job of our cell membrane is to separate your inside from your outside environment. All right? This cell membrane is semi-permeable. It regulates what gets in and out of your cell. Think of it like your house. You don't just let everybody in the front door. All right? You're able to decide who comes in and who comes out of your cell. And that's much of the case for your cell membrane. It's able to regulate what comes in and what comes out. Now, why do you separate the internal environment and the external environment? One of the reasons we do this has to do with something called compartmentation. All right? You are made of multiple cells, and we've already introduced, and I've said that you have a muscle cell, and it's different than a blood cell, and it's different than a bone cell. And the reason you compartmentalize things is so you can isolate and organize functioning. Think of it like a grocery store. Not everybody in the grocery store is doing the same thing. All right? You might have a deli department. You may have a dairy department. You may have a produce department. You have the cashiers, the checkers, the managers. Everybody, even though they may be working together for that grocery store, can isolate functioning to do different things. And that is compartmentation of your cell by your cell membrane. Now, when we talk about this in terms of being a fluid mosaic model, the fluid mosaic model describes the makeup of your cell, what it consists of. So your fluid refers to what's called the phospholipid bilayer. Your mosaic refers to all of the suspended stuff in that plasma membrane. All right? So the phospholipid bilayer is referred to as the fluid. It's a dynamic part of your cell membrane. In other words, very much like a fluid, it's mobile. All right? These phospholipid bounce around, move around. All right? And then you have things suspended within that. All right? So if we look at your phospholipid bilayer is, as the word describes, a group of phospholipids arranged in a bilayer. In other words, if I take and zoom in on this portion of it, then what that would look like is I have a phospholipid and another group of phospholipids, and they're arranged in a bilayer. Whoops. In other words, if I took them, what ends up being my black line, that group of phospholipids would continue making up that whole cell membrane. Again, a bilayer, 2 layers of phospholipids. It is these structures that we refer to as the fluid, the dynamic nature of your cell membrane. In addition to the phospholipids, though, you have a bunch of suspended stuff. Different structures embedded in your cell membrane. So the suspended stuff ends up being a couple of things. All right, when we talk about your mosaic consists of cholesterol. You have cholesterol that makes up part of your cell membrane, the mosaic portion of your cell membrane. But one of the major components is proteins. Now there's some other stuff that visible body might talk about, a glycocalyx and things like that. Cholesterol is a small portion of your cell membrane. As you go on in A&P 2, it'll talk a little bit more about that, because you have the amount of cholesterol is what can influence how readily things move in and out of your cell. But it's not a huge portion of the mosaic, probably less than about 5% of it. Proteins, on the other hand, make up a huge portion of this mosaic or cell membrane. In fact, if we added it up, around 40–45% of your cell membrane is phospholipids, and the other 40 to 45% would be different proteins, and then the rest of it's made up with the cholesterol and things like that. So the 2 major components of your cell membrane are the phospholipid bilayer and proteins. Now, your phospholipids are unique, and this is not something unique to cells or a cell membrane. It is something that's unique to phospholipids. It's in their inherent chemical nature. All right, a phospholipid, and if I drew one phospholipid here, your phospholipid has a hydrophilic head, and it has a hydrophobic tail. So hydrophilic, water, hydro, water, philic, water loving. Hydrophobic, water fearing. So when they arrange in the bilayer, the heads of your phospholipid always face the fluid environment. The tails always face inwards towards themselves, trying to isolate them away from the fluid. So what that means is the heads for this are facing your ICF or cytoplasm, and the heads from this phospholipids are facing the ECF. So the hydrophilic heads of the phospholipids face your ECF and your ICF. The tails always face inwards. All right. As I mentioned, the other major component of the membrane of the mosaic are proteins. Now, we have 2 main categories of proteins. There are 2 main categories of proteins making up that mosaic. There is what's called an integral plasma membrane protein, or from now on, I'm going to abbreviate that as IPMP. There are integral plasma membrane proteins, and integral plasma membrane proteins are proteins that are firmly embedded in your phospholipid bilayer. They tend to be transmembrane. In other words, they essentially span the entire phospholipid bilayer. I'm going to show you a picture of this in a second. All right. In general, they cannot be removed without destroying or permanently damaging the cell. All right. They cannot be removed. So if we go back to our picture here, and I have a phospholipid bilayer, then a transmembrane protein, is going to, or this integral plasma membrane protein, IPMP, spans the entire membrane. In other words, then your membrane and phospholipid bilayer would continue here, making that up. But what this means is your entire IPMP is transmembrane. It spans the entire phospholipid bilayer, which means it can associate with both the ECF and ICF. All right. Now, this is slightly different than what ends up being called our next type here. So the next category of proteins is a peripheral plasma membrane protein. All right, I'm not writing the whole bit. We'll call those a PPMP, a peripheral plasma membrane protein. All right, peripheral plasma membrane proteins are only loosely embedded in the membrane. They can be removed in general, and you don't permanently damage the cell. Now, make something else sort of clear about this. In general, your peripheral plasma membrane proteins are, in general, whoops, that's a crappy word, connected to an IPMP, but they can be on either surface. So let me draw in another IPMP and try to get you to see what I mean. Okay, come on. Over here, I may have another IPMP. Again, I would have my phospholipid bilayer continuing. What it's saying about a peripheral plasma membrane protein is they're generally connected to an IPMP and they can be on the outer surface or inner surface. In other words, what you get is maybe a PPMP that's sitting on this side or the external environment of the IPMP, or you can get a peripheral plasma membrane protein on this side. Right, they're on the periphery, either the inside or outside periphery, and oftentimes they associate with an IPMP. Okay, now this is a general category for the different types of membrane proteins that you have. All right. Here's what I sort of mean by that. Imagine I've just said transport vehicles. All right, a transport vehicle is a general term. An IPMP and a peripheral. So an IPMP or a PPMP is just a general term for the types of proteins you have. All right. So if I was to try to equate that to what I mean by like a transport vehicle, how are all the ways that you can get around in vehicles? All right, is it a truck? Is it a car? Is it a bus? All right, are all cars the same? An SUV, a sports car, a convertible, or better yet, you can get around in a boat or a plane, a semi-truck. All right, now we're getting more specific. So I could just say transport vehicles, like all the different types of proteins. All right, that's an IPMP and a PPMP. My point here is then, these then that can then be categorized. So we can take all the IPMPs and PPMPs. So you can, whoops, you can take all the, you can take all the IPMPs and PPMPs and say, what are the functional classes of those specific things? So now what I basically said is a transport vehicle, but I want to know the specific type of vehicle. Right? Is it a truck? Is it an SUV? because we know a truck does not do the same thing as an SUV or a sports car, etc. So the functional classes come then into different categories. And visible body probably talked about that as well. You can have proteins that act as channels. All right? A channel. All that may be saying is you have a type of IPMP that creates a channel to move things in and out. All right? Why would you need a channel? Well, they're selective. They allow certain things in and out. All right? But it may be because I need to move something large into my cell. I can't get through the phospholipid bilayer, so I have to have some sort of mechanism. Think of it again like your house. In general, all right, there are ways to get into your house. You come in through the window? Probably not. You use a door. And I can move furniture in through the door. I probably don't move furniture in through my window. Might not fit. So that's where channels in your cell membrane become specific, selective, if you will. I can move certain specific things in or out of my cell through a channel. In addition to channels, there are transporters. Transporters are a little different than a channel because it requires binding. You actually have to attach to it in order to bring it in. Look again back in my picture. This channel, it does not require any binding. It can just move through the channel from one side to other. If it's a transporter, on the other hand, it requires actual binding. There's usually a binding site, and it is specific to some type of molecule. It may be that we want to move glucose into your cell or some amino acid. Well, it can't just move through this open channel, all right? It has to bind. And when it binds, it's able to move things in this direction, or consequently, it can move it in the opposite direction. So a transporter is similar to a channel in that I can move large substances into my cell, but it requires actual binding. In addition to that, there are enzymes. And again, an enzyme is just a catalyst that speeds up a chemical reaction. So you have proteins that act as enzymes. In addition to enzymes, you have things that create junctions, all right? Things that connect cells together. So in other words, we can say, hold on, oops. There are proteins that act as cell junctions. In other words, now what we're doing is we're not moving things from cell to cell, we are binding cells together. So there's different types of cell junctions. All right, these cell junctions are made of proteins that link the cell together. Think of it like the thread on your shirt. All right, you have a shirt, maybe it's made of cotton, but then the sleeves are sewn on, or collar is sewn on, and we've used a thread to sew that together. The thread's not the same thing as the cotton. This is what we mean by the cell junction. There's different types of cell junctions. There are tight junctions. They are good because they actually sew, all right, cells together. So in other words, I have a cell membrane here, linked to a cell membrane here, and I have a group of cell proteins that completely fuse those cells together. That's good because they create, for the most part, an impermeable boundary. In other words, if something has to move from this side of the cell to this side of the cell, it can't go in between the cells. It actually has to go through the cell membrane, the fluid, and the cell membrane on the other side. It can't go between them. In addition to tight junctions, we we have desmosomes. Desmosomes are sometimes called anchoring junctions. They're different. They're not like sewing cells together. It's more like creating a series of rivets. In other words, if I have the cell membrane here and the cell membrane here, then basically what you have is a desmosome, and that desmosome consists of a button-like plaque. So this thing that looks like a button is a plaque. There are filaments that spread through the inside of the cell over here and the inside of the cell over here. Those are called intermediate filaments. And then there are linker filaments that link this cell and this cell. And those occur periodically. Think of them like, if I go back to your shirt example, it's not sewing it together. It's more like the buttons that are spaced out along your shirt. And what this does is it helps resist tension and tearing. You can see this really not, let me show you at home. Take a second and pinch your skin and pull it up. And I'm pretty sure, I don't care how hard you've just pulled, if you've actually done it, your skin didn't tear. Now, you know your skin's made of cells. There are cells bound to other cells, bound to other cells. But one of the things that helps those cells resist tearing, that you don't split your skin apart when you pull it, is the desmosomes. They help spread the force among the various cells. So you see this in your skin, you see this in your heart, things like that. The next type of cell junction, I got a C here. The next type is a gap junction. A gap junction is like a channel. Here's the difference. This is important. Here's the difference. If you look at a channel, way back here, in a channel, you move things from inside to outside of one cell. So we are moving from ICF to ECF or vice versa. So a channel allows you to move something in or out of the cell, a cell. A gap junction is different. All right, what actually occurs is we link IPMPs between two cells. So now if I take, this is one cell, and this is your ICF, and this is another cell, and this is your ICF, then a gap junction takes IPMPs between two cells and fuses them. Pretend there's no lines in between here. All right, I can't erase that though. All right, my point here is a gap junction connects the ICF up to adjacent cells. So now I'm able to move something from this ICF to this ICF. That will be really, really important when we start talking about muscles and you go into AMP2 and then look at a heart contraction and spreading an impulse, okay? We'll even do this when we do the nervous system, but it allows you to connect ICF to ICF, all right? So there are others. Visible Body may have talked about some of the others, but to me, those are some of the main proteins that you find making up your cell. All right, I'm not going to spend time on organelles. Visible Body does a very nice job of wandering through organelles. I will, though, hold you accountable for the various organelles. Again, you should be able to know the organelles and their general function. And that, again, with Visible Body, it should have wandered through a nucleus, the Golgi apparatus. You should have done the rough and smooth ER, and etc. Mitochondria, any of those things would be fair game. Visible Body also does a good job of wandering through transport in and out of your cell. All right. The thing, though, I want to make sure that we also understand and recognize, because it is a foundational part of A&P. It is a building block for all of A&P1 and A&P2. Probably as important as homeostasis. Your ability to maintain homeostasis revolves largely around a resting membrane potential. All right, now let's talk about what those words mean. And I'm going to try to wander through an example. When we say, let's start with the important words here. Each of those words has a meaning that then explains the overall words here. Let's start with this. When it says a membrane, what are we talking about? We're talking about your cell membrane. So the resting membrane potential refers to your cell membrane. All right, what does potential mean? Potential, the ability to do something. All right, let me use an example over here of what I mean. I think this looks good. No, it's really crappy. But anyway, this is a battery. Remember, your battery has a positive and negative side. Why do you have batteries? Why do you use batteries? They have potential. What do they have potential? They have the potential to do work. What is work? Anything that you do. Now, my battery sitting right here, is it doing anything? No, but it has the potential. All right, so my battery's in the flashlight. If my flashlight's off, my battery's not doing anything. But if I activate it, it has the potential to turn my flashlight on. Your resting membrane potential is the same thing. Even at rest, when you aren't doing anything, it has the potential, just like your battery, such that you can ask it to do something, which is work. And your resting membrane potential is so very much like a battery, because the cells of your body and the potential we're talking about is a charge. The potential is work because your cell membrane is what we call polarized. In other words, it has a charge, and it is that the outside is positive compared to the inside. All right? Your resting membrane potential, the actual charge, can vary a little bit depending on the cell we're talking about and you'll see that again as you go on to A&P. But for us, as a general rule, I tend to use your membrane is somewhere around negative 70 millivolts, meaning the inside part of the membrane is negative compared to the outside. So at rest, even when you are not doing anything, just like when your battery's not doing anything, there is a charge. And that charge is so that you can do some work when you ask it to. All right, so even at rest, when you're not doing anything, you have a resting membrane potential. And it's saying basically your cell membrane is charged. That charge is going to allow you to then do work like have a muscle contraction, send a nerve impulse, all right, communicate with other cells of your body, much like you would ask your battery to do work. This is a foundational component. All right, this resting membrane potential is largely based on ions. All right, your ions are molecules that have a positive or negative charge. All right, so you have ions, cations have a positive charge, anions have a negative charge. And we have ions inside and outside your cell. Now, I'm going to draw a cell again, and what you have to learn, what you need to know, is the ions inside versus outside of your cell. Now I'm going to start off by telling you the ions inside and outside the cell. They're the same. We have sodium inside your cell. You have sodium outside your cell. You have potassium inside your cell, potassium outside your cell. You have calcium inside your cell, and you have calcium outside your cell. You have chloride ion inside your cell, and you have chloride ion outside your cell. You also have various other anions. Those are negative charge stuff. Some books just say a bunch of different proteins with their negative charge inside versus outside. All right? So we have the same things inside versus outside. What we need to know is there's a different amount inside versus outside for these ions. All right? Now, here's what I need to do. We need to talk about a concentration gradient. A concentration gradient is the difference in amount on each side. All right? So when I say a high concentration versus a low, we're talking about the amount of ion on this side, the ICF, versus the ECF. Now, since I don't want to keep writing high and low, I'm going to put an arrow this direction that means high, and this direction that means low. All right? Fair enough so far. Okay, well, outside your cell, here's what we got. We have a high amount of sodium, we have a high amount of calcium, and we have a high amount of chloride, compared to inside your cell, where you have a lower amount of sodium, a lower amount of calcium, and a lower amount of chloride. On the outside of your cell, you have a low amount of potassium and a low amount of anions compared to inside your cell. That is something you have to inherently know. Okay? I think then visible body goes on to talk about active and passive transport. So I'm going to take this picture and I'm going to add a little bit of detail in regards to active versus passive movement of those ions. Now, these ions that you're seeing here can move across the membrane. So for all intensive purposes, these and these can move across the membrane. Anions and proteins, they don't generally move. In other words, when you think about leaking in and out, let me use sort of a different example. Go back to your house. You are running the air conditioning in your house right now, I think. And you have to keep turning it on periodically as that cold air escapes out the cracks and crevices and windows and stuff. The same thing is true of here. All right, so I'm going to try to create two lines here to try to explain what I mean. Okay, let me go down a little bit. Oops. Okay, there are two types of transport processes that visible body talked about. That's crappy. I don't want yellow. All right. There is active transport and there is passive transport. All right. Active transport. Here's what that means. It requires energy. That's my abbreviation for energy. It requires energy to move substances against their gradient. In other words, that's saying it takes energy in order to move things from low to high. Think of it like a hill. It requires energy for you to move up the hill, to roll a ball up the hill, if you will. Passive transport does not require energy, and it moves things down the gradient. In other words, let's pretend you're using energy to move a ball up the hill and then you let go of the ball. Passively, it can just roll down all by itself. That's active versus passive transport. Now, if we go back up here to my picture, I'm going to add and try to explain what happens in active versus passive transport. This is where sometimes I start losing some students in the understanding here. All right, let's just work through it and then we'll see if you have questions. Okay, to my picture here, what I have highlighted in yellow, these are the ions that readily move. Now, some move easier than others. Invisible body may have talked about how readily potassium moves or how readily sodium moves, but all of these can move across the membrane that I've highlighted in yellow. The anions and proteins, they don't regularly leak back and forth. All right. So with that being said, what I want to do is try to highlight then, and we could say, I'm going to talk about active movement, and I'll use a solid line to represent actively how you have to move sodium versus passive movement. And passive movement we'll do with a dotted line. And it basically saying, if we think of it here, how are these ions leaking? Because that's what passive is, no energy required. Passive, how are they leaking across this membrane? And then what you're using active transport for, okay? Now, let's start with passive movement, because it sort of then explains why we need active movement. Okay, so if passively I start with sodium, then which way does sodium leak passively? Well, things leak and move down their concentration gradient. They move from high to low. All right, that's easy, doesn't require any energy. And sodium, over time, would slowly leak into the cell. Now, if we allowed this to happen without any active transport, then these would equilibrate. They would end up being equal inside and outside your cell. All right, if we take potassium, potassium will passively leak this direction. And if we allow that to happen, they would equilibrate. There would be an equal amount here and there. Calcium, it passively goes and leaks this direction. Chloride also passively leaks that direction. So here's my big point. If we allowed these to all just passively leak down their concentration gradient, if we allowed all of those to leak down and not have some degree in concentration difference, there would be no resting membrane potential. There would be no charge. If you use up your battery, it loses the charge. So if these just leaked passively, then you would have no resting membrane potential. Because of active transport, you're able to maintain a gradient that then gives you that charge. So actively, because sodium is leaking in, you are constantly pumping it out. All right? Because potassium is constantly leaking out, you are pumping it in. All right? Because calcium is leaking in, you're actively pumping it out, and the same thing is true of chloride. It is that active process that maintains your concentration gradient. And because you do that, you then keep a negative versus positive charge on the inside of your cell. All right? Now, those are the main things that you should come away with in addition to all of the other stuff you learned with visible body. I'm going to pause here. Okay. That was a good bit. What I was trying to do is build on what visible body did and add some other stuff. Everybody okay? Okay. I still have some time for the next little bit. I at least want to get started with tissues because my goal, twofold. I'm going to introduce tissues. This is where it starts, the new information for what will end up being test 2. All right? Tissues is not on the exam that you have on Tuesday. So I'm going to start on tissues today, not on the exam on Tuesday, and we'll pick it back up on Thursday. Can I say the last part again about active transport? I think so. Let me see if this answers your question, because that's kind of broad. So you have resting membrane potential. It is the charge that's on your cell, meaning you have a charge of positive on the outside versus negative. All right. Now, the only way that resting membrane potential is maintained, the reason that's maintained, let me go back to sharing the screen here. The only reason that is maintained is because of active versus passive transport. All right. Now, visible body talks about active and passive transport, and they wander through and talk about what it means, a diffusion versus osmosis and tonicity, and that's all fair game, but that's a side note type thing. I was explaining active and passive transport in regards to how do you keep a charge? All right. Okay. So again, if we try to use like the air system in your house, all right, again, if you have an air conditioning unit, it runs during the summer. All right. That is because, so passively, you are cooling your house, or actively, you are cooling your house, and then passively, that cold air escapes. All right. It doesn't take any energy on you. You don't have to do anything for that cold air to escape. That's a passive process that occurs. You actively have to run your air conditioning. All right. And you know that that requires energy. Your unit's working. It costs you money. All right. So what we mean by that is, if I go back to this and talk about passive versus active transport, all right, you have a different amount of sodium and the other ions inside versus outside your cell. So what we're looking at here, pick a different color here. You have a different amount for each ion inside versus outside of the cell. All right. So here's what's going on all the time. So all the time, sodium is slowly leaking into your cell. All right. Much like we can say the air that you've cooled your house is slowly leaking out of your house. All right. But for sodium, sodium is high outside the cell. So passively, it leaks in or down its gradient. All right. So because that occurs, if all that happened was passive movement, you would have to take energy, all right, in order to then pump it out of the house, all right? If all that happened, then it would equilibrate and you would have no resting membrane potential. So you were essentially actively then pumping sodium out of your house. I can use a different example. It works better in the classroom to try to sort of get you to see it, because it's in your head. This may not seem straightforward, but this makes sense, okay? Let's pretend you have a boat sitting on the water, okay? If you have a boat sitting on the water, then hopefully you know what's the concentration gradient. Well, when I say about the concentration gradient, where's the water? Well, most of it's outside of your boat. Now, you're in your boat, you may attract a little bit of water as you're getting in there. Let's pretend you swim for a little bit, get back in the boat and all that kind of stuff, right? So there is a little bit of water in the boat, but most of the water is outside the boat. So we would say there's a high concentration of water outside your boat versus inside your boat. Now, here's why this example makes sense. Now, we've talked about letting things leak. Well, imagine I poke a hole in your boat, all right? So we've just set up, like if you can picture sodium, if you will, we've got a high concentration of water, and I now have a cell membrane, and I'm saying ions can leak across that cell membrane. So again, your cell membrane allows those ions to leak back and forth following their gradient. So if we use this boat example, your boat's sitting on the water, you got a high concentration of water outside your boat, and I've just made your boat leaky. Well, which way does water go? Well, Pat, doesn't require any energy on your part, because your boat's leaky. Water passively, all by itself, is going to leak into your boat. Now, do you want to sink? Probably not. So what do? Well, you would probably actively use your energy to start bailing water out of the boat. I want to try to keep it high outside. I don't want my boat to sink. So you have to actively pump that water back out to keep it high outside so that you don't sink. That's essentially active and passive transport for each ion. Each ion passively follows its gradient that's established by active transport. So hopefully that worked, I think. If not, just jump in and we'll work through that some more. Okay, so I'm gonna move on. We wanna introduce tissues. I'm gonna get a little bit into tissues, not too far, but let's go back to then, this is what starts the new information that will then be on test 2 and stuff. Okay. Somewhere. Okay, like I've mentioned before, again, this lecture, again, as I draw on it, add stuff to it, these will be posted in those notes and outline sections, so you'll have access to this. You can go back and watch it later and whatnot. All right, so now we need to introduce tissues. A tissue is a group of similar cells with a similar function. So a tissue is a collection of, for the most part, a same type of cell that has a job. For example, muscle tissue, what's it made of? Muscle cells. Nervous tissue, what's it made of? Nerve cells. Connective tissue, it's made of connective cells. Epithelial tissue, made of epithelial cells. So that's all that's basically introducing. And we have 4 basic tissues that you have in the body, and we're going to wander through the 4 basic types. They are epithelial, they are connective, they are muscle, and they are nervous tissue. All right. Now, as a highlight, let me mention, I'm going to wander through most of these tissues, but I will tell you firsthand, I spend the most time, at least initially, on these first 2. These first 2 are a lot of times the building block, and these are the building block for then our first organ system, which we'll introduce, which is the integument, your skin. All right, I am going to give you some basic characteristics for muscle and nervous tissue, but at least in my tissue lecture, I don't focus on these because I get you for the entire muscular system and I get you for all of the nerve systems. So we'll do fine, really good details on this as we introduce the various organ system associated with each of those. In other words, as we approach the end of the semester, we'll get into introducing the different types of muscles and how a muscle contraction works. We'll introduce the different parts of the nervous system and how they send impulses in the form of action potentials or graded potentials. So it'll seem in this tissue lecture that I'm really spending a lot of time on these 2, and it is because it's the foundation for your first organ system. And again, though, I'm not ignoring these others. We just do it mainly a little bit later. Okay. So if we look at tissues as a whole, there are 4 basic types, and we're going to start through talking about the 4 basic types as a whole can be classified. So what does it mean to be an epithelial versus a connective versus a muscle versus a nervous tissue? And part of that classification is based on a couple of things. First and foremost, the structure of the cell that makes it up. All right. Now I'm going to try to give you a very, very simple example. All right. I again mentioned muscle versus bone. Your muscle tissue is made, I don't know what I just did. Whoops, word's not responding great. I've introduced muscle tissue, a muscle cell. And what's unique about that is I think if you look at muscle, go to the store and buy some beef, some chicken, you can recognize muscle, all right? And the structure of that muscle would be very different than a connective tissue, say like a bone. But let's even look at various connective tissues. All right, I think you can all tell the difference. And these are going to be 2 types of connective tissue. I think you can all tell the difference in a bone versus your blood. And you know they're both types of connective tissue. You have a bone. It's really, really hard. You have blood. It has red blood cells in it. And so some of this classification of whether you're epithelial or connective or the different types of connective or muscle is based on the structure of the cell. All right, let's take a different example here. All right, do you know what the smallest cell in the body is? The smallest cell in the human body is a sperm. All right, that's one type of cell, smallest one in the body. And that sperm, the largest cell in the body is a female's egg that she produces. And look at how different, it's a cell, but look at how structurally different they are. If you look at a muscle cell, we can talk about a muscle cell that's basically made like a cylinder. It is very different than a nerve cell. Your nerve cell, they're some of the longest cells in the body. They can be over 1 meter long. So these cells have a structure, and that structure helps to explain how it functions. So these tissues that you see have different functions in the body. I'm going to wander through that function. Well, in addition to the structure and its function, there's also a composition of what's called your extracellular matrix. In other words, this tissue is a collection of cells, and those cells are oftentimes suspended in an external environment. And so what's that like? Well, I'm going to go back to essentially another example to try to explain what I mean. All right, I think most of you are probably familiar with your blood. Let's say that you have a blood vessel. And when I say blood, what is your blood made of? If you've ever gone to donate blood, they may be like, hey, I'm going to donate plasma. Hey, I'm going to donate blood. What do they mean by those different phrases and terms here? Well, in your blood, you have blood cells. And they may be red blood cells, they may be white blood cells, and then you have the plasma or fluid portion of it. And so what we mean here is when we talk about the cell, all right, your cell and the structure of that cell, I drew it as a circle, but I know a red blood cell is a biconcave disc. That shape, that structure of that cell helps impart its function. You'll do that as you get into ANP2. My point here is when we say the extracellular matrix, it's what your cells are suspended in. And for your blood cells are suspended in a fluid like plasma. Now, if I compared that to say like your femur, your bone, I hope that you recognize that a bone, like your humerus of your body, is made of bone cells. All right? But the extracellular matrix of it is something that's very hard. It's a hydroxyapatite. We'll talk about it later. It's very hard. So it's not plasma or fluid-like, but it's still cells suspended in some type of extracellular matrix. That's what we mean by extracellular matrix. All right. Whoops. All right. If we start with the first type of tissue, and we're going to wander through basic characteristics of epithelial tissue. Your epithelial tissue is a type of tissue that covers and lines all of your surfaces, all body surfaces. This means any cavity, any duct, whether it's exposed to the external or internal surface. All right, I'll try to explain what I mean by that. So, for example, your skin. Your skin is a type of epithelial tissue that's exposed to the outside surface. However, your stomach, the lining of your stomach is exposed to a cavity where you have your acid and all your food turning around, and its lining is made of an epithelial tissue. So epithelial tissue covers and lines all of your surfaces. In addition, all of the glands of your body are derived from epithelial tissue. So any gland, whether it's an exocrine, that would be an external, or endocrine, is a type of epithelial tissue. So where you'll see this again is when I do the integumentary, for example, a sweat gland, an oil gland, all right? The stuff that makes your ear wax, all right? Any of those things are derived from epithelial tissue. Now, your epithelial tissue differs from some of your other tissues based on its characteristics. And one of those is that your cells are tightly packed together. That means they have very little extracellular space. One of the reasons for that is because in your epithelial tissue, it is often composed of all of those cell-to-cell junctions or contacts. You'll see tight junctions, gap junctions, desmosomes, and those help to pack the cells tightly together. In other words, if we're comparing this, here's what I mean. All right, if I have a group of cells, then those cells are tightly packed so that there's very little extracellular space. All right? That's different than, say, when I look at your blood and you have blood cells that are all separated, and there is a bunch of extracellular space. Your epithelial cells oftentimes are linked by these various desmosomes. That means there's very little extracellular space. All right? One of the unique features of your epithelial tissue is that it is avascular. It doesn't contain blood vessels, so they do not have their own blood vessels. Okay? Avascular, doesn't contain its own blood supply. I'm going to come back to that thought. Even though it doesn't have its own blood supply, at least the foundational layer of your epithelial tissue has the ability to regenerate. That means it's mitogenic, or a word that goes back to your basic biology would be it can undergo the process of mitosis, cell division. All right? Now, I'm going to do these next two parts for epithelial tissue and then kind of wrap up, but it links together this avascular with surfaces and stuff. An epithelial cell has two surfaces. There's an apical surface or an apical side and a basal surface or a basal side. And there's confusion a lot of times on students' part in regards to what this means. All right, let me start with simplistically. This is referring to a side of the cell. All right, this is just the side of the cell. It's not what it's made of. Oh my goodness. And that's, again, I need to make sure that you understand this type of thing. Look, this is my phone, right? So what we're saying is it could have an apical side and a basal side, all right? You think of that as the front of my phone. I talk on this side, I can see the screen on this side and whatnot, versus that side. This is just a side, all right? Now, my point here is that's not what it's made of. The side of my cell is not what it is made of. That's what I'm about to work through here, okay? Okay, so there's an apical side and a basal side. The apical side always just means the side facing the cavity or the exposed surface. The basal surface is the side opposite the cavity or the surface. In other words, all right, let's take, for example, that you have, and let's say this is your stomach. Well, I hope you recognize that your stomach, as I've just said, this is a cavity. It would be made of cells. These may be epithelial cells. So this entire structure is made of epithelial cells. So here's what I mean. An apical side faces the cavity. So this is the apical side. This would be the basal side, the side facing the cavity versus the side opposite the cavity. In other words, it's exposed. All right? The reason we separate the side, again, as you go on in AP1 and AP2, that explains some of this, but the reason we separate that is because oftentimes, if I talk about a cell, this is one cell, then oftentimes your basal side can differ in what it looks like. So oftentimes, your apical side can look different than your basal side. You'll have some specializations, if you will. Well, it may have microvilli. Those microvilli are these like finger-like projections. The job of those, they increase surface area. So your ability to absorb food along a large surface. It may be that it has cilia, little hair-like projections that help filter and move stuff along. And this side of the cell may be very different than the basal side. But again, the apical side is always exposed to the cavity or a body surface. That is different than the basal side. Now, what I want to do now is we're going to look at on the basal side or the basal surface, there is a structure. So here where you have the basal side of the membrane, there is a structure called the basement membrane. So the basal side of the cell always has a basement membrane, and it is connecting or attaching your epithelial tissue to a connective tissue. So let's look at what that means. All right, so I can say right here, I have an epithelial cell. It has my apical side and it has my basal side. But here on this basal side, what it's saying is this epithelial cell is always connected to an underlying connective tissue. And it is right here on this basal side that you have a structure known as the basement membrane. All right, the basement membrane is the point of attachment between epithelial and connective tissue. In other words, underlying all epithelial cells and epithelial tissue, you will always have connective tissue. So underneath all epithelial tissues, you have a connective tissue. And that epithelial, that basement membrane consists of two things. Your basement membrane is an upper basal lamina. So here in A, my basement membrane, the upper basal lamina. So this that I have in green is a part of the basement membrane, and it was produced by the epithelial tissue. So this epithelial cell produces this part of the basement membrane. All right. The reticular lamina is produced by your connective tissue. So now what you have is on the basal side of the cell, on the basal side of the epithelial cell, you find a basement membrane. The basement membrane is what links or ties together your epithelial tissue with your connective tissue. And the epithelial cell produces one part of the basement membrane called the basal lamina, and your connective tissue produces the other part that's called the reticular lamina. I'm going to try to give you a different example here. Think of it like Velcro. On Velcro, you have two sections. I don't know how to explain this, but on Velcro, you have the sticky part and the part that it sticks to, and you can pull them apart, right? But there are two sides of that. That's all I'm trying to get you to see. And like Velcro, it's so that you can hold yourself together. Now, why do I bring all this up and why is it important? Here is why. If you remember, I just said that your epithelial cells and epithelial tissue is avascular. That means it does not have its own blood supply. Now, I think you would all recognize, don't you all need blood? Why do you need your blood? It carries oxygen. You need oxygen. And what I'm sitting here saying is your epithelial cells do not have their very own blood supply. They are avascular. Well, that means it doesn't have a blood supply within it that directly supplies it with oxygen. So where does it get its blood supply? Well, the blood or the oxygen that you need for that epithelial cell to be able to survive occurs because your connective tissue has blood vessels. Now, these blood vessels do not go up into the epithelial cells, but what ends up happening is, because if you recognize back from basic biology, how does your oxygen and carbon dioxide get in and out of your blood vessels? It occurs by diffusion. So this is avascular but its blood and oxygen comes through the process of diffusion from the connective tissue. And that is the importance of this basement membrane holding them together. Imagine, if you will, that instead, imagine you had an epithelial cell and the connective tissue is way over here. Well, if this is your blood supply, then via diffusion, it would have to diffuse across here and way across here to get, and that would take a lot of time. It would be like you're suffocating yourself. All right. So part of the importance of this basement membrane is it links your epithelial cell together. That's both for support as well as then to get its oxygen via diffusion. All right. That's a good foundation. That's a good place to start for tissues, not get too far along in tissues. I should be able to maybe wrap that up on Thursday. Okay, so a couple of things. You've got a test on Tuesday, not lecturing on Tuesday, but your first exam's Tuesday. It'll be due by the end of the day, Wednesday. Before Sunday of this week, you have a Blackboard quiz. It's on your Blackboard shell under that content area, under exams and quizzes, 10 points, 10 questions. And you have now, I'm going to open here before afternoon, a visible body graded quiz. Again, the graded quiz on both Blackboard and visible body, you only get one opportunity to take it. Again, if you're well prepared, I think you shouldn't have too much trouble. All right, I'll stop the recording there.